presentation, we're talking about the synthesis of antioxidant modified P188. My name is Platte, and these are my colleagues.
I'm Hayden, Fredericks.
Discaya.
So, cancer biology. Approximately 50% of cancer patients use radiation therapy as a form of treatment throughout their diagnosis. So radiation therapy is high-speed particles that damage the cancer cell DNA and kill or prevent replication. Although radiation is so highly used, there are some drawbacks to this. It can cause inflammation and chronic oxidative stress, which can be classified as DNA modification, changes in protein shape, and cell membrane degradation.
Okay, so our group is mainly focused on the cell membrane degradation of molecules that affect due to cancer radiation. So you may be wondering why we're modifying P188. Well, P188 is a poloxamer. It's actually a triblock copolymer, as you can see above. And it is composed of a central 27-unit hydrophobic chain, which is flanked by two 80-unit hydrophilic chains. And the hydrophobic chain is polypropylene oxide. The hydrophilic chains are polyethylene oxide. So specifically, P188 has been explored as a very promising reagent to modulate and aid in cell membrane healing. It has been shown to afford an enhanced structural stability to cell membranes, and it also has been shown to help have cell membrane resealing properties. So you may be wondering how P188 is actually capable of resealing holes in the cell membrane. Well, it is not entirely understood. However, P188's amphipathic nature, or its composition of both hydrophobic and hydrophilic subunits, is hypothesized to be responsible for facilitating the cell membrane resealing. Lee and coworkers observed that in relation to a lipid monolayer, the hydrophobic subunit of P188 actually inserts into the acyl lipid portion of the lipid monolayer, which is also hydrophobic. And then the hydrophilic arms, PEO arms of P188, associate themselves with the hydrophilic extracellular environment. So based on this observation, it's hypothesized that P188 would have a similar interaction with cell membranes, which are lipid bilayers, and act as a so-called molecular band-aid to help cell membrane resealing. So, as mentioned previously, ionizing radiation exposure does generate reactive oxygen species, which can alter cell physiology. Specifically, when free radicals interact with our cells, they frequently cause cell membrane permeabilization, which are holes in the cell membrane, and this could lead to cellular death or cellular necrosis. As a proposed therapeutic approach, there has been the use of utilizing the addition of antioxidant cofactors to increase the effectiveness of P188. And this is because antioxidants have a capacity for free radical scavenging, and this allows them to bind to and neutralize those reactive oxygen species that are caused by the radiation. And you can see on the side of our screen, these are some of the antioxidants that we are specifically targeting with this research, which include lipoic acid, ferrocene carboxylic acid, and gallic acid. So because of these antioxidants' ability to neutralize these reactive oxygen species, this should increase cell viability, and this therapeutic will aid in the mitigation of radiation therapy side effects.
So, considering that P188 can reseal membranes, we thought and the previous discussion on antioxidants, we thought perhaps there's a way we can modify P188 so that it can not only reseal cell membranes, but also help reduce the amount of harmful reactive oxygen species out there. So our proposal was, what if we attach antioxidants as head groups to P188 to improve the cell membrane healing properties of P188?
So using a modified synthetic procedure from Zhang, I attached lipoic acid, or LPA, to P188 using DMAP as a nucleophilic catalyst, DCC as the coupling reagent, and then THF as the solvent. And this experiment produced a 53% yield with a greater than 95% functionalization of lipoic acid on the terminus of P188. And so in order to determine if this reaction was successful and we synthesized our target molecule, we employed proton NMR spectroscopy. So this is a tool that allows us to assign resonance values, which are these values below, that correspond with the number of protons that are in our target molecule. So the NMR data is consistent with lipoic acid's successful attachment to P188, and our target molecule is shown above. In this NMR, the protons that are depicted in purple correspond to the lipoic acid protons, and the protons that are depicted in red correspond to the protons of P188. So additionally, these two much larger resonances associate with the JK and LM protons of our main P188 backbone structure, and then the N protons over there are associated with the methyl group in our P188 backbone structure. And we know that because N should have 3 protons attached to that carbon, and there are 27 units, that should have an integration to around 81 protons. So because the methyl group is easily isolated, and we know this, we chose that to help aid in our analysis. And we are very pleased with the integration values of the protons coming from lipoic acid and P188. As you can see, the protons that are integrating with that methyl group below are around, they're 81.02. So in general, NMR data we say has around a 5% error, so we conclude that this product has a 95% functionalization of lipoic acid on the terminus of P188.
So I mainly worked on ferrocene carboxylic acid with P188, and this procedure right here was based off a similar procedure, a synthesized procedure by a former colleague, Nadia Ferguson, where I used DCC as a coupling reagent, DMAP as a nucleophilic catalyst, and DCM as my solvent. And while I haven't obtained full characterization of my product yet, a similar experiment with similar set of conditions had shown their product to reach 65% functionalization. And while this isn't 100%, we are making efforts to improve on this.
So for my reaction, I am using gallic acid to flank P188. As you can see, I am starting with a protected gallic acid, and this will just help my reaction just be a little bit quicker. I'm using EDC as my coupling agent, and then the DMAP is my catalyst. Once I will get my product, I will change the aryl groups and hydrolyze them and turn them into hydroxyl groups.
So as for our future directions, well, one of our future directions will be looking at other antioxidants, such as DOPAC or tannic acid. Additionally, we are looking at other ways to modify P188 to expand the range of possible antioxidants that we can attach. Additionally, I'd like to mention that we have other collaborators on this project, such as Dr. Tim Hanks and his team at Furman, who are working on the molecular modeling and characterization of our products, and Dr. Yost and his team at MUSC, who are working on the biological testing of these products. And well, relatively recently, his team had a model undergoing cellular stress, and they showed that with lipoic acid flanked P188, these cells had more cell viability compared to just P188. Overall, though, the project intends to work with the goal of creating a therapeutic treatment that can be used in conjunction with radiation therapy to mitigate its harmful side effects.
So we would like to thank NSF, Furman University, MUSC, Dr. Robert Harris for allowing us to participate in this research project, and our other research colleagues who are not here today, Grace Lindsay, Kate Johnson, and Nadia Ferguson, for their work on this project, and also our collaborator, Dr. Tim Hanks, and his students for their work in molecular modeling and characterization, and Dr. Yost and his team at MUSC for their work in biological testing. Thank you.
All right, do we have any questions for this group?
I'm not very familiar with antioxidants, but the antioxidants that you tested, are they commonly found in humans or are naturally present in the humans, or would this be something that's newly introduced to the body?
So in terms of what we're working with right now, some of ours, I guess it kind of depends on what antioxidant we're talking about. In the past, we have used ascorbic acid, which is vitamin C, which we can find in our body. But we specifically chose these antioxidants because we know that their reactions won't cause the creation of any harmful side products. So it would be something that we're adding to the body, but they're all antioxidants that are FDA approved, so there would be no risk associated with them.
What about vitamin D? Could you use that one?
So specifically right now, we're targeting antioxidants that we can use for esterification reactions. So all of our antioxidants have, if you see like that similar side group, it's a carboxylic acid. So specifically, we were doing an ether attachment when we were looking at ascorbic acid, but it was getting a little bit difficult because of all of the other hydroxyl groups that are associated with it, along with Kate's reaction that she's working on. That's also why we're starting out using a protected version, so that specifically the carboxylic acid is what is going to be reacting with P188 as opposed to one of the other hydroxyl groups. So, but that could be very interesting for us in the future to look into.
So if the main goal for like reducing the trauma that occurs to the body with radiation is like kind of reducing these reactive oxygen species, have you ever thought about maybe just like increasing the NADPH and just like pairing with the natural metabolism that goes through the body, which that might be totally out of nowhere, but just like in biochem, you know, they talk about how NADPH is like a huge reactive oxygen species like producer.
Yes. So because like I haven't looked into any of that research specifically, but with what we're doing, like obviously radiation therapy generates so many reactive oxygen species that our body cannot deal with it. And so that's why our normal cellular functioning for helping like cell membrane resealing can't reseal those cells in the correct amount of time for all of the other reactive oxygen species that are within the body at that same time. So specifically with adding the antioxidants, we'll kind of be like killing two birds with one stone, because we'll have our antioxidants attached to the end of P188. So that will be reacting and binding with any other reactive oxygen species within our body that aren't interacting with other cells, while at the same time it will also be inserting into the cell membrane to help reseal any harmful effects that have already been caused by reactive oxygen species.